plant growth. After we've covered the plant, at least an intro to the plant cells and tissues idea, we'll come back to some of that when we cover root stems and leaves, but now we're getting into plant growth. And in order to consider plant growth, we have to think about hormones because hormones regulate a lot of what's going on with plant growth. So what I'm doing in this little section is just giving you an overview. If you take plant physiology, we go into more detail. This is more like an introduction to the idea of plant hormones and some of the things that are regulated by them. I think we left off here with the idea of there's a group of chemicals that the plant makes that are called auxins, and they were the first discovered. Do you remember who discovered it? Darwin, yeah, Charles Darwin and his son Francis. And so they didn't name it. The name came later, but they're credited with figuring this out and getting a start on it anyways. And so we left off here with the idea that these achenes are actually dry fruits that cover the surface of this lovely strawberry. And so the dry fruits produce auxins. And so they were able to demonstrate that auxins are regulating the development of the rest of the strawberry by picking all the achenes off and watching what would happen. And the growth of the strawberry was arrested and stopped. And then they said, well, what happens if we pick all the achenes off and then spray it with an auxin solution? And when they did that, the strawberry pretty much formed almost close to normal. And so that was a pretty simple experiment, but it was able to show that the presence of auxins are necessary to get the fruit development. And of course, they've gone on since then, and they have confirmed that that happens with a lot of different fruits. Another thing that it is famous for is this idea of apical dominance. Apical dominance. So apical dominance, now we're zooming into the apical bud, which is at the shoot tip, okay? And excuse me, this is going back to Charles Darwin's experiments where they were thinking there's something in the shoot tip, some kind of chemical that is triggering the plant to bend toward unidirectional light. And so they now know that auxins are present in the shoot tip. And what happens is it will course down the length of the flow, down the length of the stem. And as it's doing that, it will cause the apical buds. So the apical buds, I mean, that's interesting. Okay. Whoops. Yeah. So here, the lateral bud, it's a little messy here, the way they have it. You have your apical bud and then your lateral bud. Lateral buds are all these little spots here where you see a leaf coming off. And those are another area of active growth. When the shoot tip, though, is actively growing, auxin is produced there. It starts flowing down the stem, and it essentially tells those lateral buds, hey, just chill out, be quiet, you don't need to grow. Okay. I want to also write down another word for lateral bud is axillary. You'll see that too, probably. So I want to give you that term. So axillary buds are the same thing as lateral buds. And so those are dormant for long stretches of time. If that auxin is flowing down the stem saying, hey, just be dormant, you don't have to do anything right now. So on the left picture, what you're seeing here is they don't have the apical bud fully in the picture, but the apical bud is intact. And so the auxin is flowing down and it's keeping these right here, those are the axillary or lateral buds. It's keeping those just dormant, they're not doing much. Then they say, what happens if you amputate? So right here, they've amputated off a big chunk of the stem. So definitely they amputated the apical bud. All right, so that's the amputation right there. And then what happens? Those axillary buds that were just little bumps of tissue, now they're starting to grow. right? So now you have that release. Now those axillary buds are, because there's no oxen telling them to be quiet anymore, they're like, okay, we're gonna grow. And so they start to go through cell division and grow, giving rise to more leaves. So this is a common strategy that they do if your plant's getting what they call leggy, it's getting too tall and it's kind of spindly looking, you can nip off the axillary or the apical bud to get a more bushy appearance, more contained plant.
Is auxin a hormone then?
Yes, it's a hormone that's made, a lot of it you can see in the apical meristem. It's found to other places too. So that's what's called apical dominance. If the shoot apex is intact, it's growing, auxin's there, it starts to flow down the stem, and it's dominating the growth. It's the apical stem that's dominating, you know, the shoot tip is dominating. And so they learned that one pretty good early on, and that's a nice trick you can do with your houseplants. If they start to get too, oh, there it is, axillary bud. If they get too leggy. Often plants will get too leggy, by the way, if you're not providing enough sunlight or light. And that, unfortunately, the house I live in was not designed by anyone who thought about plants, because the windows face the wrong directions. The south facing one is into the garage. It's like, come on. And so my plants don't get enough light. And so they tend to get leggy unless I'm providing some extra augmented light for them. So. Okay, another thing that auxins are famous for, that actually should be an S on the auxin, because it's a group of many different types of chemicals that act as auxin hormones, is this idea of adventitious roots. So these three pictures are showing different versions of adventitious roots. So here you have a like an orchid, and you have the adventitious roots coming off the stem. Here is it's like a stem cutting. Someone took a stem cutting and put it into some media, some gelled media, and are starting to launch the adventitious roots. Here it looks like an ivy plant. Ivy stems tend to have these adventitious roots that allow them to, very often they cling really nicely to like brick walls. You might have seen that on older houses where they have ivy covering the house. So adventitious roots are just roots that are made not from the original root tissue in the seed. All right, so we have the radicle coming out of the seeds that you planted on Thursday, right? That's like true embryonic root tissue. Adventitious roots are not that. Yes.
So, since I the ability to
Yeah.
how are they able to like control which one they do?
It's often concentration dependent. So there's like for auxins, there's a sweet spot that really promotes growth. And then if you go on either side of that, it tends to, it can start to inhibit. And we do those types of experiments in plant fids where we'll kind of do a dose dependent experiment. You can see that impact where too much and it starts to slow down the effect. In this case, it'd be growth. So adventitious roots, if you've ever taken, like if you've been at your friend's house and they have a lovely plant and you're like, oh, can I take a cutting of your plant? And then you put it into some water on your windowsill and watch it form these adventitious roots. So what's happening is actually amazing, because the stem tissue, when you take a cutting of your friend's plant and you put the stem into some water and let it be for a while, you'll start to see these adventitious roots. And what's so amazing about that is those are stem cells, you know, that was stem tissue. And all of a sudden now they're shifting gears and becoming root tissue. I mean, we can't do that. If we cut off a finger, we can't even grow back, let alone, you know, decide to grow a toe instead of the finger. So here, what they do, those cells, right at the cut area, those cells are pretty much shifting gears tremendously. Their gene expression shifts. They're like, we don't have any roots anymore. And so they like, we got to make roots. And so they'll shift their gene expression and start forming root tissue. And so that's this idea of adventitious roots. Okay, it's not coming from the original root tissue of the seed, you know, the baby plant inside the seed. And so you'll do that. We're going to do a propagation experiment soon. Is it this week? Next week, where you go into the greenhouse and you'll be propagating some plants there. And so you'll be looking for these adventitious roots. And so if you buy, Root Tone, I think is one of the brands. You can buy it in like a gel or a powder. And they say buy this if you're trying to get your plant to root really well. The powder is a mixture of auxins that are in that. So I think we'll have some in the lab provided to you if you want to dip your stem cutting in that before you put it into the water. That'll help a little bit. So they're really good at doing that. And the other really important thing that they found out about auxins is in general, they stimulate cell growth and they're very necessary if you're going to clone plants using tissue culture techniques. And so tissue culture is the idea of using artificial media to clone your plant. So it's pretty amazing that you have these plant cells. You can take a little chunk of plant cells and put it onto the right, like agar media with some auxins mixed into it and some other things in there. And that will stimulate that little chunk of plant tissue to divide, grow. And if you aren't providing all the different hormones, if you're just starting with auxins, it tends to, first of all, form like a clump of cells. It almost looks like a tumor, but we call it a callus in the botany world. It's called a callus. And that's just a mass of undifferentiated plant cells. And then what you do is you mix in another hormone and transfer this into another media that has another hormone, which I'll talk about in a second. And then that tells those cells, okay, start to produce roots, start to produce shoots, start to produce leaves. And so you get this nice differentiation. So without understanding that, I mean, tissue culture, we just wouldn't have been able to do that. And now tissue culture technique is a big thing. That's how they grow up lots and lots of plants. If you like African violets, for example, that's one of my favorite houseplants. Most of that's done through cloning and using tissue culture techniques. And so many plants are grown that way for business, to sell them off to people who love plants. So very, very important. I'll get into phototropism and gravitropism too. So tropism, that idea, that part of the word is kind of like movement towards, or movement, I guess you could say. And so photo is light, and of course gravity. And so let me come back to that, but they know that auxins are involved in those two mechanisms, which I'll explain in just a few minutes, okay? All right, so that's an intro to the auxins. Another group that's very famous are the gibberellins. Again, many different chemicals fall under this category of gibberellins. So this is another hormone that's naturally made. This picture is showing their claim to fame. When they first discovered gibberellins, they realized it causes tremendous growth in the stem. So here you're seeing the plants below the ladder here. That's how they normally look. They're called dwarf plants, dwarf cabbage. But if you spray the plant with a solution of gibberellins, it causes this massive growth like this. And so they're like, what is going on here? They actually discovered it in rice plants. What happened was, they were growing rice out in the rice paddy, and they noticed their rice plants were getting too tall. And you don't want that, because then what happens is they were getting too tall too quickly. And when they do that, what happens is they grow tall too quickly and they don't really reinforce their stem tissue that well. And so they get a little floppy. And what happens then, and you can imagine, as the rice grains start to mature on the top of the stem, it gets heavy. Gravity starts to pull, and the rice plants bend over and end up, the grain is in the water of the rice paddy. Well, that's not what you want. That's terrible, because now the grain is in the water It's starting to get attacked by fungi. It's very difficult to harvest. It's a disaster. And so they wanted to know what's going on. So they went into the rice paddy, like, why are these plants doing this? They never did it before. And they studied what's going on in the field, and they found it was actually a fungus, a pathogen attacking the rice plants, causing this. So the fungus was actually exuding out gibberellins, and the plant was responding to it, going, oh, I guess I better grow. And so from the fungus perspective, pretty cool technique, because it got the plant to fall over essentially, and now it has all this access to the lovely grain, and it just ate it all up. So from the fungal perspective, it was a good strategy. Of course, from the people who want to harvest the rice, terrible. So they actually found it in the fungus, and they're like, huh, what the heck is this thing that the plant's responding to? They characterized it and said it was a gibberellin. And then they started looking for it in the plant, and they're like, oh, the plant makes this naturally, except it, in this case, the fungus was making too much of it, and it really caused the rice plant to overgrow in its length. So we do this in the plant physiology lab with pea plants. We have dwarf peas. You can buy them. They're dwarf because they have a mutation, a really tiny mutation that causes like 1/20th the amount of gibberellic acid to be made. So they don't make very much, so they stay short. And then if you spray them with gibberellic acid, they'll respond to it. They have the ability to respond. They just don't make very much of it. So after you spray them, they can really shoot up in their stem height. So it's kind of fun to play with in the lab. And there's several different like dwarf plants like that they make gibberellic acid. It's just a small amount. So one of the things that they use gibberellic acid for in food industry is actually to make bigger grapes. And what's really happening is they spray the vine, the grapevine, and it's the vine tissue that starts to elongate. So basically what happens is there's more physical space in between the grapes. And so they have more room to grow big and juicy and sweet, which is what we like. We'll pay good money for that rather than small tart grapes, right? I mean, we like the big, sweet, juicy ones. And so that's a common strategy when they're growing grapes, either for just direct eating or wine production. They'll spray them.
Is this hormone, why we have GMOs now? For food production?
I don't know, because they're not genetically modifying it. They're just spraying it. And the plant has everything it already needs to respond to it.
Mhm.
So they didn't have to genetically modify it. So that's one nice thing. You don't have to worry about any of the GMO issue. So they also know that gibberellins are in the seed. And when the seed is getting certain cues that it's time to germinate, then gibberellic acid will also kick in, and its production goes up in the seed, which is another, clue to the seed. Okay, let's get going. So let's see. You put seeds in wet paper towel, right, on Thursday? Can you think, or did you go over any of what is a seed paying attention to when it decides it has to germinate? What could be one thing?
Fall.
Pardon?
Fall.
Thaw?
Yeah.
Yeah, it going through a cold cycle, right? In fact, if you've ever bought seeds and you see on the packet, it has to be, what's the word? Not frozen, there's a fancy word. Stratified, there we go. It has to be stratified, they say, and that usually means you wet the seeds and put them in your freezer or your fridge and let them go for a couple months or so. And then they think they went through winter. It's a nice trick. So that, a lot of seeds have to do something like that, but not all. What did you do to the seeds to get them going? Get them wet. That's a huge clue. Because seeds are pretty dehydrated. They need to dehydrate so that fungi don't attack them. Because if you have a nice wet seed, fungi think it's food and they're going to go after it. If the seed is really dehydrated, they can kind of get away with hiding away, so to speak, from other things that want to eat it. So wet the seeds, that's a huge clue. Going through a cold period, that's another one. Sometimes light, you might see packets where they say sprinkle on the surface of the soil. Because light has to, like lettuce is like that. And then the other things they don't really ever say on a seed packet is, once you start to wet the seed, you will promote this idea. The gibberellic acid is in the seed, it gets the clue to start ramping up its concentration, and it will start to promote the breakdown of some of the carbohydrates, especially in the seed, to help the seed start to get food for itself, because initially the seed has to provide the nutrition because the plant's not doing photosynthesis yet. So there's usually enough food production or food stored away. And then after that, the very first thing that comes out, what's the name of that? Again? A radical, which is the embryonic root. That's the true root tissue. Seeking water because it's so dehydrated, it needs water. So the other thing they've noticed is that gibberellins are involved in the life cycle of the plant when it has to shift into essentially going through puberty. Plants have their own version of puberty. And so we don't really say puberty in the botany world. We tend to say it has a juvenile phase where they don't produce any reproductive structures that are workable. And then they'll mature into the adult phase. And so to get that transition, you need to have gibberellic acid involved in order for that to successfully happen. Yes.
Does it increasing?
Yes, in this case, yeah. Mhm. So very, very important. There's other things that gibberellins do, but those are some of the kind of the famous things. Cytokinins are another group of hormones. Cytokinins. So this may look vaguely familiar if you took BI 111 recently and had to go through mitosis. Remember that? Mitosis and meiosis. So this is the process of mitosis. And I put this picture here because cytokinins are heavily involved in the process of cell division. So in mitosis, they're involved in that for the plant. And in fact, I think I said last week that they can't make a plant that is devoid of auxins. They also can't make a plant that's devoid of cytokinins. They can't create a mutant that can't produce the cytokinins. The plant just dies. And so those two groups of hormones, the auxins and cytokinins, are so critical for the plant in terms of cell division, cell growth. And so one of the bigger areas of the plant is the root tips, where you see production of the cytokinins. But you can find them in really young parts of the plant, like baby leaves that are just starting to grow and develop, the beginning of fruit formation, the embryo inside the seed. And so again, you can't create mutants of cytokinin deficient plants. It just doesn't happen. They also promote the axillary or lateral bud growth. They help promote that. And they're necessary to get the chloroplasts, that organelle, to fully form and be up and running. Pretty important. If the plant doesn't have really good, robust chloroplasts, it can't do photosynthesis correctly. So really very critical. So again, you can start to see why it's hard to produce a plant that can't produce cytokinins. It just dies because it's so important. So with cytokinins, one of the things that is also pretty critical. So when you have a plant going through its life cycle, and of course, you've witnessed this, especially if you've lived in the northern realms where we have seasons, the beautiful fall colors is a very visual representation of the plant going through senescence. So that's just a fancy word that is referring to like the natural cell death of of the plant. It could be just some of the cells, or it could be, in this case, whole, all the leaves are going through senescence. Now, what happens in the fall is that these leaves are starting to turn these beautiful colors, primarily because a lot of the chlorophyll is getting siphoned down into the root for storage. And all the lovely nutrients that are in the leaves, first, all of that gets siphoned down into the roots. And then you start to see the other pigments that are there, and they start to show up. And so that's senescence. Well, cytokinins slow that process down. And so you can even do experiments with, trees if you have trees in your backyard, if you want to have an experiment, spray one of your trees with a cytokinin solution and keep the other one just going and see what happens. You should see a delay where in this picture here, what you're seeing is on the right, the plant went through senescence. Same identical plant, they sprayed it with cytokinin, and you can see that it's delaying senescence. The leaves are still lovely green, still kicking along with photosynthesis. So cytokinins are kind of regulating that phase of the plant and are able to slow it down. And we take advantage of that sometimes, especially in the food production industry. So it's an interesting process. Okay, here's one that's just one chemical. Abscisic acid is just abscisic acid. It's not a group of different chemicals. So abscisic acid, think of it like a brake on your car, the brake pedal. It has inhibitory effects on stimulatory effects produced by other hormones. All right, so it kind of makes sense that a plant has to have both a gas pedal and a brake pedal when it comes to regulating its development and growth. You can't necessarily always be growing. Sometimes you have to say, okay, enough. And so having a brake is a good idea. And so abscisic acid is like that. It's a good analogy. And what it does is, one of the key things that makes a lot of sense when you think about it for a minute is that it makes sure the seeds don't germinate too early. Okay, and so if you think about angiosperm, the flowering plants that produce, like an apple tree, and it's going to produce the apple fruit. You wouldn't want the seeds in the apple already germinating, because what has to happen is you want that apple to ripen, get visually appealing, have a fruit disperser get it, and then hopefully they'll defecate out the seeds somewhere else away from the parent plant. And so you want the seed quiet and dormant through all of that so that it won't germinate until it's away and into new territory and habitat. And so that's a pretty important process for the plant to make sure the seeds stay dormant. So the other thing that's happening to your seeds right now that you put into wet paper towel, that wetness is actually diluting the abscisic acid now that's in your seeds. It's diluting it down so that the gibberellic acid now is ramping up. And so that switch is what's causing your seeds to start to wake up and go through germination.
How about like the types of I guess like a bell pepper, when you open it, sometimes there's like a little baby of pepper inside. Is that because the acid kind of failed?
Yep. And you can see that every once in a blue moon, right? And so basically the abscisic acid didn't do its job. There wasn't probably enough of it in that area, in that tissue. So yeah, abscisic acid is really interesting it also is keeping all the leaf buds on the trees out here. It's keeping it dormant. Abscisic acid is there keeping it dormant because of course you don't want tender leaves coming out on the maple trees right now. They'd be toast. Well, they'd freeze to death, not really toast, but so they maintain bud dormancy, seed dormancy, really, really important aspect of the plant's life. You need to make sure there's a break, especially in areas like this where we have long, cold winters. So abscisic acid is very important. It also has another really important function, Do you know what we're looking at here? Stomata. Very good, very good. So I think you can tell this one has a bigger opening over the pore compared to this one, right? So if you are a plant, imagine you're a plant and you have your roots in the soil and the soil starts to go through drought. And so it's getting drier and drier and your roots are like, oh boy, it's getting dry. I think I mentioned this last week too, what's happening is, so the roots are the first to figure out we're under a drought. But it's really the stomata that have to be controlled now, because what happens the minute you open your stomata? What starts to leave your body? Water. So if the roots are like, oh boy, we're in a drought and the stomata are way up in the leaves, you got to get the message there, right? You got to tell the stomata, hey, we got an issue here. So what's happening is the couple of things. Abscisic acid will stimulate roots to grow because they're searching, they're foraging. Those roots have to forage for water. And they are starting to grow, maybe try to get deeper, and they're searching for water. So they'll actually forage through growth for water. That's huge. So that's really good. But then the other thing they need to do is tell those stomata, you know what, you might want to close down at least for a while here. And let's just see how this goes. And so what happens is abscisic acid is stimulating root growth, but it's also getting sent up the plant to the leaves, and then it reaches the stomata and it causes them to close down. So that's what the one on the right, it's a little bit more closed down. And so plants have the ability to really have a fine tune what the percentage is the pore open. Is it 100% open, 50%, 10%, 7%, 13%? I mean, they can be very, very fine tuned on this. And again, if you know that as soon as you open your stomata, you're losing water, now it makes sense. Okay, you have to tell the stomata to close down. What's the drawback though? Let's say it's daytime and the roots are saying, sending abscisic acid, hey, close down. What's the drawback? Good thing is you have water staying in you, but what can't you get access to? Yeah, that your respiration slows down, definitely, for sure. But what else can't you get? Can't do photosynthesis. You can't get in the CO2 you need. So now you're sacrificing growth. And it's a trade-off. But plants will do that if they have to for a while. And they can do it for a good stretch. And then hopefully a rainstorm will come through and they'll be able to reverse it. And so it makes really good sense that plants have really tight control on what's going on with their guard cells. And there's actually more to it than what I'm describing. It's not just abscisic acid. They're also very sensitive to light and a certain quality of light. They actually pay attention to blue light. When the sun is striking the plant body, that sun can be broken down into different wavelengths, right? You did the experiment when it passes through a glass prism and it breaks into the rainbow colors. And so sunlight has blue quality of light, and that's actually what's causing the stomata to open in the morning. It's actually responding to that blue light, like, oh, it's sunrise. It's time to go ahead and open up. Okay, so that's hugely important for the plant. They have to be able to assess water amount in their soil system and then potentially tell the stomata to close down. Okay, another hormone that's just a single chemical is ethylene. This is a gas. It's actually a gas, ethylene. And so out of all the hormones we've discussed so far, this is the only one that's a gas. And it is famous really for being a, so unfortunately, abscisic acid, that hormone was nicknamed the stress hormone, which with water stress, yes, but really is ethylene. That's kind of like a general stress hormone. And ethylene is produced in a lot of different areas of the plant. You can see it being produced in the fruits, flowers, seeds, leaves, and roots. It's pretty much all over. It also can get triggered, like it has a feedback mechanism where if you have ethylene getting made, it actually stimulates, make more, make more, make more. So it's kind of got this feedback loop going where you can ramp up ethylene amounts pretty quickly in the plant. So this is very important if you grow food because you want to be able to time when you're gonna harvest your fruits to send to market. And so one of the big things that ethylene can do is ripen fruits. They know it's heavily involved in getting that transition from unripe to ripe. And then you can, harvest and send it off to be purchased. So they've known that many fruits respond well if you expose them to ethylene, and it'll cause them to ripen up. And so they use that in the food industry. The other thing that it can do, which is kind of almost like, well, it can cause what they call leaf abscission. Abscission means to fall off. The leaf stem will fall off the plant. So I don't know if we've introduced this term yet. When you look at, it's supposed to be a P. I don't know why it disappears when I write down low on the slide. It will. Look at that. Why? Look at that. It's like magic. Very annoying magic. All right, I'm going to write it up here. See, now it works. What's up with that? I don't get it. No matter how many times I try that, if I go down low like that, it won't do it. Petiole, have you been introduced? Can't remember if we talked about that. That's the leaf stem essentially. So here's the leaf blade, a holly leaf blade. And then the petiole is like a little stem. So that's the area we're zooming into, where the petiole attaches to the plant. It will go through abscission and it will create this. So if you have your petiole, this is the petiole, and then you have oh my goodness, come on. Is it going to make that disappear? So it's got a very fat petiole there. And then here's the stem that's attaching to. What's going to happen is right in that area where it's attaching, there'll be like, hey, it did it. It upsized. It'll create this zone of the cell walls weaken in like a straight line right where it's attached to the stem. And that weakening in that zone causes the petiole and the leaf blade to fall off. So that's leaf abscission. So here they did an experiment where they put two holly plants and they inverted a beaker over them. And one is just the control here. This one has an apple inside. Why did they put the beaker over it? Keep the gas. Yeah, keep the gas around the plant. All right. So what they did was, I wish they would have showed, if you cut into the apple or damage it in some way, it immediately starts releasing ethylene. And so now ethylene is getting released, it's gaseous, and it's creating those abscission zones on the petiole. And you can see the poor holly plant is now devoid of leaves. So anytime you bite into an apple, or you take a stem cutting, or you rip your leaves for your salad, you are stimulating ethylene production because you're damaging that tissue. So ethylene gets produced in response to damage. So that's why this apple, if you cut it or, slam it on and bruise it, it's going to start to release ethylene.
Why would you put in a bag of like some sort of, you see there's like a rotten one, this rotten ones?
Yes. Yes, I have that going on right now. I had a batch of apples I picked. Yeah, and the top layer, they all look good, you know, and then I dug under, it's like, oh, no, and that's what happened there. Pardon?
Yeah, that Corey.
I know, right? What, you know, what's that saying? One bad apple spoils the bunch. It's true. And now you know what? It's the ethylene getting produced. So yes, you gotta be careful about that.
It actually produces gas like storage in itself, or is it like something it's taking in?
It actually synthesizes it.
Okay.
It's very complicated pathway. It's called the Yang cycle. It's named after Dr. Yang. And yeah, it's an interesting, long biosynthetic pathway. But, you know, so if, that's why if you, well, no, I won't get into that. But basically, anytime you wound a plant, whether, even if it's a, you know, you want to take a nice stem cutting in your, it's still right where you cut it, you have ethylene getting produced. So can you imagine if you own a flower shop? Have you gone into a flower shop and you go into the cooler area and there's all those cut stems of roses, carnations? They got some issues to take care of, right? I mean, those are wounded plants. You're selling wounded plants. I mean, they're beautiful, but you have to make sure your ethylene levels are low in that refrigerator. So,
You rent it then, or?
they can exhaust it out. There's certain chemicals you can add into, they're usually, the stems are usually in a bucket of water. There's chemicals you can put into the water that either stops ethylene biosynthesis or it blocks the receptor. So ethylene can't do what it's supposed to do.
So like, if you bring it flowers from the store and get that like little food packet, is that like the
It
reserve?
stops ethylene. That's why you should put the food in. I think they label it something cute like plant food or something. But one of the ingredients in there is to stop ethylene. And so you really should use it because it'll make the flowers stay nicer longer. Because as soon as you cut those beautiful roses off the rose bush, it's producing ethylene. It's on its way to senescing and dying. And so I had a good friend who used to own a flower shop and we chat about this quite a bit because, you know, that's your livelihood on the line. You don't want to sell a rose or, you know, a bunch of flowers and it's dead in a couple days. You won't get that person coming back. They're saying, you know, they'll say your plants die too quick. So yeah, it's very significant for if you're trying to make a living with cut flowers. Well, there's, I don't know if this will answer your question, but there's some fruits that, let me show you this next slide. I don't know if this is really what you're going for. So like apples will produce ethylene if you cut into them, especially wound them. Bananas can respond to external ethylene. So what I'm showing here is if you've ever gone to the store and all you have available to purchase are green bananas, isn't that annoying? You're like, I want a banana tomorrow for breakfast. I don't want to eat a green one. You could try this trick. You could buy the bunch and put the cut apple and then close it, either put it under a big, you know, bucket over it or in a bag or something to trap the ethylene, and then it'll ripen. So they know that some fruits will ripen with exposure to ethylene from an outside source. Whether or not different plants or different fruits produce different, probably, but I don't know for sure. The short answer is I don't know, but I would guess that to be the case. And some fruits can respond this way and some can't. Some will like, no, it's not going to do anything for me. And so they actually have lists of which fruits can respond. And it happens that bananas can. They're called climacteric versus non-climacteric, which you don't need to know that. But. So yeah, try that next time and see if it works for you. But ethylene is. So here's the cut flower idea. So really, I thought, you know, unfortunately, abscisic acid got the claim to fame as stress hormone, but really it's water stress. Ethylene reacts to almost any other stress. If the plant gets too cold, ethylene gets produced. If it gets too waterlogged, if like a flooding event, ethylene gets produced. I mean, pretty much any time a plant is getting stressed, you'll get ethylene. being made and ramped up in the plant. So, and on top of that, auxins and cytokinins can stimulate ethylene production. So they can link in with each other. And they know that ethylene speeds up senescence, all right? So this is the cut flower idea where certainly wouldn't want this to happen to your cut flowers. And so this STS, that's a chemical that you can add into the water and it will slow down the production of ethylene. And so this one had that in its water here where the cut stems could take it up. You see the lovely carnations. This one did not have that. And so after 14 days, you can see the difference. So ethylene can kind of speed up the death process. Okay. Any questions on any of that? I like how you ask questions during the lecture. I really like that. I think that's important because if you have a question, it's probably somebody else has a similar one too. Yes.
Can you explain again why the auxins, cytokines, and like
Yeah, this is something that I give a lot of credit to people who study plant hormones because how do you tease this apart? I mean, how did they figure this out? But basically, like the whole idea of leaf excision, I didn't get into it, but it's like a three-step process. As fall starts to happen, the temps, the light changes, and the light length changes, what you see happening is auxin production in the leaf goes down, cytokinin goes down. And that actually causes ethylene to go up. And so what I'm trying to say is you get these interactions between the hormones in it and trigger, in this case, trigger ethylene. Basically, the leaf, if it starts to lose auxin and cytokinin, that's telling that. I like this stuff. We can go ahead and, cycle the nutrients down and I'll fall on. Any other questions? Okay, so the other thing we have to talk about with this slide set is plant. So that's like an intro to the hormones. Now we're gonna look at like plant growth overall and how they might respond to their environment. And I'm also gonna try a Google Forms thing on Friday as well. So bear with me on that. We're gonna see if it works. All right, so we're gonna try that on Friday as well. So we'll see you then.
Thanks so much.
Yeah.